This project was created for Anthropology 205, Biological Anthropology. My name is Sabrina Mohammed, and today we will discuss the topic of protein synthesis, a cooking analogy. Protein synthesis is like cooking a dish with a recipe, the recipe being the gene and the dish being the protein. And there are 2 steps in that process. Transcription is step 1, which is the copying of the gene, and translation is step 2, the building of the protein. Think of DNA as the ultimate cookbook for all the recipes, aka your genes. And DNA can't leave the nucleus because genes are very important and they must be kept protected. The DNA code consists of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. mRNA is like a recipe card that is used once, then discarded. It's when mRNA copies a gene code from DNA and transports outside the nucleus because it can leave. This process of copying the recipe, the code, is called transcription. We have uracil replacing thymine in this process, only in the RNA codons. After copying down our recipe, we get to make our dish, our protein. Now we go into the kitchen, the cytoplasm, to make our dish, the protein, in a process called translation, made by our chef, the ribosome. The ribosome reads the code and then generates the right protein from the reading of that code. The ribosome attaches to the mRNA and begins to read the code. The ribosome doesn't read the code 1 letter at a time. It reads it in groups of 3. Each group of 3 bases on the mRNA is called a codon. These codons are the specific instructions for which amino acids, our ingredients, to use next. The tRNA is like a delivery truck that delivers the right amino acid to the right codon. Each tRNA molecule is perfectly matched to carry one specific amino acid ingredient. On one end of the tRNA is the amino acid, and on the other end is the anticodon, which is the 3-base sequence that perfectly matches a specific mRNA codon. As the ribosome chef moves along the mRNA recipe card, a tRNA delivery truck arrives, dropping off its ingredients, the amino acid, at the exact spot indicated by the codon. The ribosome then forms a strong peptide bond to link the new amino acid to the growing chain. The empty tRNA trunk is discarded and the process repeats with the next codon. The ribosome continues this assembly line process, moving down the mRNA, reading codon after codon, and connecting amino acid after amino acid. This continues until the ribosome hits a stop codon, the signal to stop cooking. The result is a long linear chain of hundreds or even thousands of connected amino acids, called a polypeptide chain. This chain is not yet the final dish. When the chain is complete, it immediately begins to fold and twist itself into a complex, unique 3-dimensional structure, our protein. This specific shape is our dish. This protein can be active and functional as an enzyme to digest food, a hormone to send a signal, or a structural piece to build the cell. I hope you all learned how to make a new dish today, learned about the DNA cookbook, and the mRNA recipe card that copies a recipe from our cookbook, and our delivery truck, the tRNA that helps us mix our ingredients for our final dish, a protein. Thank you for listening, and goodbye.